Hello, my name is Aaron, and I'll be presenting about the gut microbiome in which my group and I presented on. Well, first, you have to ask yourself, what is a microbiome? Well, a microbiome is just the amount of bacteria and organisms living in a group or living in an area at one time. So, specific to this project was the gut microbiome, which was the amount, the proportion, and the types of bacteria living within the stomach and some portions of the small intestine. So here you can see the abstract in which we typed and then our introduction as well. So the significance of a microbiome and the significance of us having bacteria within our gut is so our diet, health, and all the other stuff could be up to par. For example, if there is any alterations or if there is any imbalance within the bacteria within our gut, then then health factors such as gastric cancer and irritable bowel syndrome, as well as obesity, start to come into play. And as we all know, that's not what we would like. So this then allows us to raise a question of how does the size of an organism affect the types of microorganisms within the host? Our hypothesis specifically was the amount and the strength of microorganisms within the host increases as the host itself increases. The reason why we chose that hypothesis was because different size organisms equals different amounts of bacteria. If a large organism is larger than a small organism, for example, a fish, then we would automatically assume, or my group automatically assumed, that the microorganisms within the larger organism has to be more and stronger because of the size of that animal opposed to the fish. So now on to our materials and method. So the animals in which we used two dogs, a pig, and a hedgehog. So we compared all of those. We did all the experiments, or we ran through each and every procedure in which we did. But at the very end, we kind of compared two different dogs. But that I'll get to later. So we started off in week one. In week one, what did we do? We isolated DNA, and specifically the tool that we used to isolate the DNA was the QIAGEN kit, or the QIAGEN kit. And then after we isolated that DNA, and within week one of our first day doing this experiment, we also concentrated the DNA in which we isolated prior using the Zymo kit. And we concentrated tenfold, meaning that we concentrated it 10 times more than it originally was. So then going on to week two, the significance of week two was we used ultraviolet absorption spectrometry in order to determine the concentration and to see if there were any proteins or any contaminating factors within the DNA that we isolated. So after we did that, we also determined, or we also used background knowledge, knowing that DNA is read at 260 nanometers, and then protein is best read at 280 nanometers. So from then on, we go up into within week two, we also amplified the specific DNA, and we amplified using the PCR reactions. And using the PCR reactions, we took 3 steps. First step was to denature the DNA, and the denaturation of the DNA, we had to get the temperature to 92 degrees Celsius, and after, or 95, sorry, and then after we got it to 95 degrees Celsius, the DNA was denatured. From then, we had to lower the temperature to 52 degrees in order to let the primers bind to that DNA. After that took place, we raised the temperature back up to 72 degrees in order to allow the process of the DNA, in order for the DNA to start replicating. And with that replication, we made millions of copies of that DNA. And the amount of cycles that we did for this were 25 cycles, thus producing millions of copies. Then on to week three. The significance of week three was we used gel electrophoresis And the significance of this gel electrophoresis was to determine to see was another way to see if we got product from this DNA. So as we all know, the PCR cleanup worked. So using that gel electrophoresis, we determined that the PCR product did work because we saw that there were bands present in the positive, not the negative, but present in the positive in all of our samples. So then after that, we removed the unused primers, and after removing the unused primers, we ran that through again. And after we ran that through again, we ran it through for 3 minutes, and we kind of did the same cycle. So 3 minutes, 95 degrees Celsius, dropped it down to 52 degrees Celsius or 55 degrees Celsius, and then raised it back up to 72 degrees Celsius. And we did that for 5 minutes, and then we held it at 4 degrees Celsius. So then moving on to week 4, we performed the PCR cleanup once again. And we did that in order to determine the concentration of the amplified samples that we used in the fluorometric assay. So this was more sensitive than UV spectrometry because this allows us to determine if, it allows us to determine specifically DNA, if DNA was present, RNA, or any other protein and stuff within that solution. So the DNA sequencing was also done by using the Illumina MySequence Next Gen DNA Analyzer. So that was pretty much all of how we went about conducting our experiment. So now, like I mentioned, this was our gel electrophoresis. So our gel electrophoresis indicated the positive had a band, the negative did not have a band, but the hedgehog, pig, and both dogs also had bands, and they were all around 550 base pairs. So moving on to the results, you can also see that our Amplicon PCR, we had our hedgehog, pig, 2 dogs, the sample numbers, and the Amplicon PCR quantification numbers, and these numbers were read and were successful. So using the nano drop, we determined the concentration of each organism, and as you can see, for the hedgehog, it's 26.3 nanograms per microliter. Pig is 8.8 nanograms per microliter. The dog 1, dog 2, and so on. And then the ratio is very important in determining whether, how successful we were in producing these the concentrations. So with that being said, since our readings were between 1.5 and 2, which is this ratio, all of our readings were between that, thus implying that we were successful in getting product and isolating the DNA and everything that we did in the previous weeks. So now on to our kingdom, order, genus. As you can see, we didn't really use the organisms that we tested within the weeks that we've been working with. Instead, we used 2 different organisms, which were a flat-coated retriever and a husky. So we indicated a sample name. The kingdom was 99% 99% for the flat-coated retriever, then 92% for the order, and then 82.41% for the genus. And the reason for this is the higher the percent for the kingdom is supposed to be 95% or greater, and seeing that it was shows the amount of bacteria and how successful that was. But then the husky is a little bit different. It's a little bit lower than it should be, but that just shows the percents of each. Now on to the total number of reads. So the total number of reads that we got that we analyzed for the flat-coated retriever were 18,028, but the number of successful reads were 17,852. Using math, we can already assume that the number of successful reads compared to the number of unsuccessful reads were greater. So that determines how successful they were in determining the amount of bacteria within this. And then the husky, we also got a large amount of successful reads as well, seeing that we got 1,850. So then, finally, another thing that I want to indicate about the taxonomy was that the percent of unclassified reads will increase as the taxonomy levels also become more specific. So this means that seeing that we only looked at kingdom, order, and genus, that's why the percents are so high. But if we were to continue going lower and lower and lower, that's getting more specific into each specific organism, and thus the number of unclassified organisms are going to increase, because since each organism is different, each organism uses different things to break down whatever food that they have in their gut. So, for example, people or organisms living in Antarctica opposed to organisms living in Africa most likely have two different types of most likely are in different species, thus meaning that the organisms and microorganisms within their tests are going to be different. So now on to the discussion. So the discussion, well, we pretty much, our interpretation of our UV absorbance data, it was pretty much successful. And the reason why that they were successful was that we had high concentrations, like I said before, because our readings were between 1.5 and 2. So now our data shows that we also, like I said before, had a high concentration for the PCR cleanup. But now, now back to the gel electrophoresis, it's just pretty much we were also successful again because we had bands, bright bands in all of the categories, including positive, but the one category that we didn't have a band in was the negative, and the negative we didn't have a band in because we used water instead of actual DNA, and thus no band came up, and no band was supposed to come up. So now we are on to the number of reads, successful reads compared to each animal, and it is adequate. And the reason why it's adequate is because of the amount of successful reads that we got compared to the amount of total reads that we got. So that proportion is significant in seeing if we were successful in obtaining the amount of successful reads that we were supposed to. And then finally, clearly addressing the whole experiment. Going back to our question, does the size of the or does the size of the organism affect the types and amount of organisms within each host? Our hypothesis was indeed supported because, for one, when we analyzed the flat coat and the husky, the flat coat is larger than the husky. And we can see that the percents for the flat-coated retriever are greater than the percents for the husky, and the same thing with the amount of reads for the flat-coated retriever opposed to the husky. The flat-coated retriever had greater values for pretty much everything compared to the husky. And then going to the animals that we conducted ourselves within week 1 and between week 1 and week 4, we saw that the two dogs had a higher concentration. And the higher concentration meaning the higher concentration of data when we finish isolating and quantifying and also concentrating each piece of DNA. We saw that the dogs was greater. Dog 1 had 30.9 nanograms per microliter, and then dog 2 had 26.5, which are the greatest within this chart. So just going back to everything, this pretty much supports our experiment supports our hypothesis that the amount and the strength of microorganisms in each host does increase as the host size also increases. So finally, as we all know, since I didn't come up with any of this research myself, we have a works cited right here, and our works cited just includes some of the academic sources that we use in order to come up with some of the background knowledge, and also our lab manuals. It's not included in here, but we also use a lab manual in order to follow the steps accurately, gain a little bit of background knowledge, and to conduct the rest of this experiment. So thank you.